Okay, let's have a class. Let's talk about the calculation. Okay, that will be part of your exam questions. So please take the notes. Let's talk about the calculation. That will be our last lab. Okay, that's the microbial. Microbials in foods. And I already saw the results, looks pretty good. Okay, let's give some example. So Victoria, you group, can you please count the ground beef? How many colonies are there? We just gave you the example. If you have a ground beef, then you do the same thing. Okay, that's a ground beef. The second one, we're gonna have a hot dog. Who has a hot dog? You can go ahead and count. Okay, then the third one, what we have is tomato. So who has a beautiful tomato plates, you can go ahead and count. And last one, we have a chicken. Chicken carcasses. The calculation is important, so I want you guys to understand. Okay, let's do that. Who has tomato? How many you have?
26.
Oh, really good, that's. So we're gonna see the 26, which is, we say CFU on the plates. And here we just calculate everything. Okay, okay, we're not gonna really focus on that range, because that's what it is, Can you tell me the dilution factor of the tomato? What's the plates you have? The 26. What's the dilution factor?
Yeah, 10 to the first.
Okay, 10 to the first. Then what we have? Then how you do the calculation is 26 multiplied by 10 to the first. That's dilution factor. And then remember how many solution we added there? 100. Is that right? We add 100 ml. So what you get is 26 multiplied by 10 to the 3. That equals 2.6 multiplied by 10 to the 4. That is CFU per tomato. Which means the whole tomato, how many bacteria are there. And we also could transfer to the log. If we become a log 10, how many are you going to get? We're going to get approximately 4.1 log 10 CFU per tomato. So this is what we did. Okay. Now 10 to the first, this is we call it a dilution factor. So that's the tomato. Who had ground beef?
64.
That's really nice, the range for 64. Okay, the 64. Not sure you may not can see it, so I'm going to use this one again. Okay, 64, how we do it? What's the dilution factor?
10 to the first.
10 to the first, so what we do is the same thing. 64 multiplied by 10 to the 1. And remember how we did it. We were doing is 100 ml, 0.1% buffered peptide would add 25 gram of tomato, of the ground beef, sorry. Then we divided by 25 because they all coming from that 25 ground beef. And this section equals 5. Therefore, we got 64 multiplied by 10 multiplied by 5. That equals 220 multiplied by 10. That equals 3.2 multiplied by 10 to the 3. How many it is? If you change to log, that's almost 3.3 log 10 CFU per gram. But remember, this is what you count is a big colony. There's a lots of tiny colony there. So almost this ground beef have natural flora is about like 4 log or even 5 log. So you can see the ground beef, there's a lots of microorganisms there. Why? Because there is a grounding process. You have utensils, you have those grinders, and you also touch the packaging. That's a more complicated procedure. They sometimes will generate those mild heat and even favor the bacteria to survive. So that's ground beef. If you did the test today, you will be feeling there's a lots of bacteria there, and you're never gonna eat it if you were not well cooked. Because these could be E. coli, could be fecal contaminated. So that's why it is a target for E. coli 0157H7 and the non-0157 Shigella toxin, generally E. coli, because you see the overall microbial quality is not that nice. Okay, tomato makes sense because tomato coming from the soil, so that's why it's also higher. Okay, let's do hot dog. Who has hot dog? Can I have the number for hot dog? It won't be 2 minutes. Okay, you get 5. Okay. So if you get 5, what you do? What's the dilution factor? 10 to the 1? Oh. 10 to the 5th?
Yeah.
Then how about you place of 10 to the 1 and 10 to the 3?
I don't have any.
Okay, that means it's possibly you switch to the dilution plates, which means you should go the other side.
Oh, yeah.
How many you have?
We had 8 on 10 to the 3rd.
Okay, that means kind of like a switch there. So let's just do average, okay. Divided by 2 equals what? 6.5. Okay, let's just assuming, okay, let's just say you do the 10 to the 3. Then what you got? 6.5 multiplied by 10 to the 3. You have to multiply by 100 and divided by 50, because that's 50 square centimeter and that's ml. So this equals 2. Then this whole thing equals 13 multiplied by 10 to the 3, and that equals 1.3 multiplied by 10 to the 4. That's approximately 4.0 log 10 CFU per square centimeter. Okay, that is basically the hot dog. We just gave you example if you have 5 and 8. This looks fine, but I believe so. It should be much lower than that. It should be, if it's 10 to the 1, you only got 1 log there. So you can look at your plates. Your hot dog most likely looks good, because hot dog we have antimicrobials there. So usually it's not very high, and also it's well cooked. Okay, chicken carcasses, what you have? Who has chicken carcasses? How many? 8? Okay, no, not bad. Who else?
31.
Is it hen or chicken carcass? Hen.
It's a whole chicken,
Oh, chicken, okay, 31.
not the.
Okay, now let's do an average, 31. Divided by 2. That equals how many? 39. 19.5. Okay, so what we did is 19.5 multiplied by 200. Okay, that's equal. How many it is? This is roughly CFU per ml. We call it a rinse rate. Yeah. What's the dilution factor of that one, by the way? 10 to the 1? Then you have to multiply by 10 right here. Okay, so that calculation have to be changed. Sorry. And that calculation has to be changed. I made a mistake here. 10 to the 1, don't worry about that. We'll just do 19.5 multiplied by 10, that equals 195 CFU per ml. That's a rinse rate. And this, if you change to the log, it's approximately 2 log 10 CFU per ml. That looks okay, because chicken, we have a very strict processing. in the United States. We'll talk about that in the lecture, okay, in about an hour. So this is the calculation. And in the final exam, we'll have some questions talk about that. So please be familiar with the calculation, how we do it. Okay, we have reviewed it again. Ground beef. This is the final dilution, the colony on the plates, dilution factor, and this will be how we do it, because 25 gram mixed with 100 ml, where it comes from, it comes from that 25 gram ground beef. And tomato, we do the colony on the plates, the dilution factor, and this is a solution we're using 100 ml. So that is all the bacteria coming from the tomato. That's why we say per tomato. And the hot dog, this is the same thing. We are doing is square centimeter because that's the surface usually. And chicken carcasses, we don't care how many it is. We will just do is a per ml rinse rate because the chicken carcass is very large. And it's geometrically it's not uniform. That's why we just do. And here the results relatively make sense. And we see the chicken carcasses is really low because we have a very strict cleaning sanitation and standard procedures to the post-harvesting of the birds in the United States. So that's really good results. Okay, so I'll move to here. Then we're going to come to our review section. We're going to talk about from the beginning, after the midterm exam, what we have. So we had a big chunk of the section here is biochemistry test. And the biochemical test. So we're going to have to review every single one for you again. And which one positive, which one negative. The first thing first is 3-tube fermentation test. The 3-tube fermentation test, if you remember, we had a 3-tube. The 3-tube looks like is a king cap tube. Inside we have another small tube. So what we have is inside we have another small tube there. And what we have, these are the sugar. We have a glucose, dextrose, and sucrose. Sometimes we can use lactose, okay? When the bacteria during the fermentation, they could generate gas and generate acids. For example, lactic acid. Now, how do we know there is an acid generate? That turns yellow color. If it turns yellow color, we know it is acid generate because there is a pH indicator called phenol red. The phenol red turns yellow at low pH. Okay. Yellow at low pH. And you have to remember this line, what we draw here is, if it's low pH, pH like 2, it is yellow. pH right in the middle, this is the original color. And if it's pH very alkaline, let's say 12, this is we call it a cherry red. Or a little bit of pink, a little bit red. So that's a phenol red. Now how do we know there is a gas generator? So gas generator, when you go here, you will see this Durand tube. On the top, there is a bubble. There is a gas bubble generator. That means the gas comes out. Now, which bacteria is actually is a fermentation positive? What we did is E. coli. It is fermentation positive. It is gas positive, and also it is acid positive. Okay, that's a fermentation test, is the first one. Second one, what we did is urea test. So that's a urease test. So we're gonna go review a little bit urease. It is NH2C carbonyl group NH2, if bacteria has ureas, will break down, become ammonium and carbon dioxide, and this carbon dioxide is going to go out, comes out. So what happened is, in this medium, we also has a pH indicator, which is phenol red, but at this moment, this phenol red turns a little bit cherry red. So this phenol red here, which is actually turns cherry red, or a little bit pink, we say. Because the ammonia comes out, and it will be fit right here. It's a small alkali, okay? And which bacteria is positive? It's a citrobacter is positive. And it is a surrogate. We talk about the pathogen called Proteus vulgaris. Now in the final exam, it's the same thing as before. All the bacteria name, you don't have to remember how to spelling. At the beginning of the exam paper, I will all write there. So you pick from there if you need it. Okay, that's the second test, is urea test. First one, sweet tube fermentation test. Okay, now the third one. Third one, what we did is SIM test. And this is, we say, it's a one shot, 3 drops, and we need to use is a needle instead of a loop. So the first thing, what is this? Sulfide. Okay, how do you know it is sulfide positive? The whole thing turns black. Because there is iron ingredients there. The iron ingredients will be reduced and then turn black. And what does I means? That is indole. Okay, what is the indole? That's a byproduct of tryptophan. And if you want to draw it again, if you forgot about it, since it's an exam, you're finished. So lots of you forgot about it. C, this is N, NH2COOH. Is that right, that tryptophan?
No, that's wrong. No, you're good.
Yeah. No, not good. Something missing right there.
Yeah.
You have NH2. Yeah. So that's tryptophan. And the tryptophan, bacteria could use it. How they do it? We go from here. And this part, we call it an indole. Okay. This section is indole. So we draw, we don't test the structure here. Okay. I just make sure you know it's indole. And how do we know the indole is positive? I add a big drop of KOVAC, that's called Kovac. And this Kovac turns cherry red. And that is indole positive. And which bacteria is indole positive? And I always say it is E. coli. It's a very quick test for E. coli. Is indole positive. And which bacteria is sulfate positive? This is Citrobacter. Okay, what is the M in the last one? This M has to be careful. Here is motility. So remember how we did that? We had a tube which is a semi-solid agar because we had only 0.5% agar there. And we wanted to see whether bacteria stay in the middle or goes everywhere. So some of the bacteria rather stay in the middle, there's no motility. If they goes everywhere, which means there's a motility positive. And what we did is Citrobacter, which is motility test positive. Is positive. Because it's a surrogate of Proteus vulgarius, and that is Petrotrichus. Okay, so that's a very important test for the SIM test. So when we have this SIM test, we're following to do another one, phenylalanine deaminase test. Phenylalanine deaminase test. So phenylalanine deaminase test, remember how we did that? Is a phenylalanine slants. We add a bacteria there, we do the incubation at 35 degrees Celsius 24 hours. Now how do we know it is positive? We were adding a reagent, is ferric chloride. And we find that it turns dark green color. That is phenylalanine deaminase test positive. Now why it is important, if you want to draw the structure again, we'll see this looks like a phenyl group. So you have a CH2, so you go over here C, NH2, COOH, you have the H. And once you have a deaminase, which means this guy is gone, that's called a deaminase. Then the left side here, this whole thing is very magic stuff. We call it phenyl pyruvate. Because this is called a modified TCA cycle. Okay, and this is part of the deaminase is gone, so this is deaminase. And that means the bacteria could use amino acids as a nutrition ingredient, same as the Indo test. So which bacteria is deaminase test positive? Is Citrobacter. In other words, Proteus vulgarius is positive. Okay, so deaminase test. When we finish this one, we have something, other things to do is a catalase test. And this is relatively easy for the catalase test. So we have the hydrogen peroxide. If bacteria has catalase, what they're going to do, they're going to become water and oxygen, and they're showing you the bubble. So this is very easy, is you have a glass slides, remember? And we put a bacteria there, we mixed it up, and then we add a drop of the H2O2, and then when you see the bubbling, it is positive. Now, I said this test is very important. Why? Because clinically, this is differentiate the Staphylococcus versus Streptococcus. Okay, Staphylococcus should be catalase tested positive, and the Streptococcus is catalase tested negative. That's what you did for the throat culture, remember? And the throat culture, you have to do is a candle jaw. And why we need a candle jaw? Because we want to generate a microaerophilic environment, which means there's approximately 5 to 10% of the oxygen, and they have almost 15% of the carbon dioxide. That's favorable, which is a better hemolytic pinpoint colony. To grow. Okay, that's catalase test. Then the catalase test, I have a very similar one we want to mention. Here's oxidase test. So how we did the oxidase test, remember I asked you guys to do is agar plates. On the left side, we adding citrobacter, and on the other side, we just add some other bacteria, let's say E. coli. And then what we do, we're gonna dump in the reagent called oxidase reagent. And when we find it's just area turns purple color, that means it is positive. So citrobacter is positive. E. coli here is negative. That's the oxidase test. Oxidase test, clinically, it is to differentiate Pseudomonas and versus other bacteria, which is we call it Enterobacteria ACA. And the reason is these group of the bacteria, they are all gram-negative, non-indostar forming. And it is, we call facultative, and it is gas fermentation positive, and this is also acid fermentation positive. And when we do a McConkey agar, we see it is a pink colony. So we use McConkey agar. It is pink colony. So it is very difficult to differentiate. That's why we can using Pseudomonas and Enterobacteria ACA. Pseudomonas is also positive for oxidase test. And this is negative. That's why we could use another tool for the enterobacteria to do the analysis, which is we call it an enterotube. Enterotube 2. Remember that beautiful pencil you took it to the home? That's enterotube 2 for the multiple channel testing. Okay? Not done yet, we have some other tests. And more VC test. This is one we have to be careful. AEM VC test. What's the difference between that? What is I? That's Indo. What is M? This is methyl red. Be careful. And I'm going to be to do this. Will be circle this, okay? This is methyl red. That's not motility. What is V? You can just say VP, okay, Vogel's first here. What is C? Citrate. And what's a VP for? Which is testing whether your fermented products is acid or neutralized. But the more important here is we're going to differentiate E. coli and Enterobacter. What we got, E. coli is Indo positive, methyl red positive, VP negative, citrate negative, and the other guy is negative, negative, positive, and positive. This is the group is important because it's like a sticky ends, restriction enzyme like. Okay. So E. coli, Enterobacter, the IMVC test. When we finished this test, we had a group of tests, if you remember, which means a bacteria could use other resource besides we called about the glucose. What will we do so? Remember, three agar very similar. We all did is split them onto two, and there is a two plates on one side. we inoculated Bacillus subtilis. On the other side, we are adding E. coli. It's a very similar situation. This is BS, Bacillus subtilis. On the other side, we add E. coli. And the last one, I said the little bit difference. This is we add Staphylococcus aureus. And also the right side, we add E. coli. Now, what is these tests for? The first one is the skin milk agar. So that is skin milk agar. The skin milk agar, we are trying to see whether there is a transparent zone. If they have a transparent zone surrounded, what this means, which means they're going to break down the protein, which is caseinase. So it is caseinase test positive. Always just say protease test positive. Okay, so Bacillus subtilis is positive. And the second thing, if you remember, this is starch agar. And starch agar, we need to see whether bacteria has amylase. And which bacteria has it? Is Bacillus subtilis is positive. But how we did it? We not really for transparent zone. We gotta have to dumping gram iodine. Is that right? When the iodine is showing up the brownish color, which means there's still starch there, so it's amylase negative. So iodine, we say if it's brownish color, then no amylase. If this is no brownish, then that means there is an amylase. Amylase test positive. Okay, the last one, what we have here is the split brew agar. Split brew agar. And this one, simply we see transparent zone, but we didn't do very good because most of you, the culture looks like the agar plate looks like the blue is faded. But ideally, the Staphylococcus aureus is positive because they have a transparent zone, and this is used to test lipase. And lipase test positive. So that's number 8. Okay, that's biochemistry test where we finished so many. And then we have a clinical test. What's a clinical test? Well, we did that. Clinical test, we use the urine test, UTI. Okay, which bacteria is UTI? E. coli, it's simple. Number 1 cause a pathogen. And what's the symptoms of UTI? Called dysuria, which means it's difficult to pee. And we have different steps to do. We need using a 2 agar, which is McConkey agar, and a blood agar to do a qualitative test. Okay, if this is a positive, this is also usually in the positive growing there. That's a qualitative test. Qualitative test. But more important, so we are using a nutrient agar to do the quantitative test. That's quantitative test. If you see a colony is more than 100,000 CFU per ml, and remember what we did is a loop. The loop surface, we have a very tiny catching the liquid. This is 0.001 ml, so the dilution factor is 1,000. And that's clinically diagnosed as a UTI, 100,000 CFU per ml. Now we also could do a test which is a dip stick test. What's a dipstick test for? Look at 3 things. Number 1, white blood cell. Number 2, red blood cell. Number 3, nitrite. Now, white blood cell, we look at the leukocyte esterase. Yeah, we not really look at the white blood cell. So that's a clinical test for UTI. Well, we have the last one. Aerobic respiration. Last one is the pound water testing. Okay, this is the one what we should know. I tested lots of you the time. You should always remember. We have a plate, agar plate cell. It's a paper petrofilm, paper agar. So when you see these dots there, what are these? That's APC, aerobic plate counts. Aerobic plate counts. Okay, here, you see those red dots there? That is coliform. But more important, you should see there is some of the purplish, blue purplish colony there, and with gas, that's generic E. coli. And when they add up them together, that becomes total coliform. Okay? Antibiotic testing will be an extra points this semester because the bomb threatening, some section didn't do. So be very fair. Okay, but do you still remember how we did that? I'm gonna be add this on the top. The last one will be the extra points. So what we do, we had a dipstick there. So let's say tetracycline, 30, let's say ampicillin, 10, for example, we see the transparent zone going to surround it. So what we have to do, we use a ruler to measure the transparent zone. Remember, it's millimeter, it's not inch. And then we're going to compare to the table. We'll see resistant, intermediate, and susceptible. And this one we call it susceptible. Not really say it's sensitive. Okay, because that means bacteria choose a reaction to the pathogen. Pathogen reacted to the antibiotics, not for the human being. Okay, so this is overview. The final exam of the outline. Final exam will be Thursday after Thanksgiving holiday. So Tuesday you have time to review. Do not forget, if you want an insurance policy, what you should do. During the final exam, you should bring your lab notebook. Now you need to bring when you're taking the exam. You cannot bring afterwards. You rent a car from Enterprise, you're going to have to buy the insurance first. Can you hit the car already, have an accident, then you come back to car, said, I need to buy insurance? No, it doesn't work, is that right? So it does not work here. That's what we say, lab notebook is an insurance policy and 20 points. So, in other words, AEM 341 lab is not a difficult class. You should at least, the majority of you will get A or A minus. Majority of. Because we have that insurance policy, but you have to do it. You don't have to. If you're very confident, you don't have to. Like I said, this antibiotic resistance test will be the extra points because be fair to lab 3, lab 4, they didn't do. And but if you need that insurance policy, that's good. Now, last thing, it needs to be a book, a lab notebook. This is called a book. This is called couple piece of paper. You know what I'm saying? You can say you need 10 points and give me a couple pieces of paper. And what we need is a book. Like you buy insurance, you either buy a liability, you either buy a full coverage. Can you just say I'm gonna buy half liability, which means I hit the top of my car, the front of my car, I got the insurance. If the bottom, it's okay. No, that means you need to have a book. Okay, so because why we want to do that, I always say the lab notebook is the best thing to study, to review. So work with your bench partner, or even you can have a study group. If some of you miss some of the labs, then you could, using this time to go back to finish your lab notebook. You get the insurance points, at the same time you review all these material. So that's what I encourage. Now you certainly can do this during the Thanksgiving, after Thanksgiving holiday, Monday, Tuesday, Wednesday. You can do that. Have a 3 days and just reorganize your lab notebook. If somebody keep doing it, it's great. If you didn't start to do, that's good. And also my students, Carly Long, will have a review section Wednesday. And if you need help, please come in. Okay? So that's all we have.